Hello everyone, welcome to the next lecture in our physiology video lecture series. This particular lecture is titled Motor and Integrative Neurophysiology. This is the second lecture following the sensory neurophysiology lecture before and will complete our lectures on the nervous system. So let us begin. First, let us look into the motor functions of the spinal cord which control the cord reflex. Sensory information is integrated at all levels of the nervous system and causes appropriate motor responses that begin in the spinal cord with relatively simple muscle reflexes, extend into the brainstem with more complicated responses, and finally extend to the cerebrum where the most complicated muscle skills are controlled. Hence, there is a hierarchy from the spinal cord, the brainstem, and to the cerebrum, depending on the complexity of the motor control required for the reflex. This is similar to what we have already seen in the sensory neurophysiology part, where there is also a hierarchy of the spinal cord, the hindbrain, and the cortex. Without the special neuronal circuits of the cord, even the most complex motor control systems in the brain would not cause any purposeful muscle movement. For example, there is no neuronal circuit anywhere in the brain that causes the specific to and fro movement of the legs that is required in walking. Instead, the circuits for these movements are in the cord, and the brain simply sends command signals to the spinal cord to set into motion the walking process. The brain thus gives directions that control the sequential cord activities to promote turning movements when they are required, to lean the body forward during acceleration, to change the movements from walking to jumping as needed, and to monitor continuously and control equilibrium. All this is done through analytical and command signals generated in the brain. But it also requires the many neuronal circuits of the spinal cord that are the objects of the command. Hence, essentially, the relation between the brain and the spinal cord can be compared to a musician and his piano. Without the musician's help and direction, the piano can't play the beautiful piece of music that it is meant to. But without the piano itself, the musician also cannot generate a symphony. The relation between the brain and the spinal cord is similar. Brain commands the spinal cord nervous circuits in order to generate movement, but without the spinal cord nervous circuits, the brain cannot generate any purposeful movement. But even without the brain, the spinal cord circuits themselves are not capable to generate complicated movements. Now, next, let us look into organization of the spinal cord for motor function. The figure shows the typical organization of the cord gray matter in a single cord segment. Sensory signals enter the cord almost entirely through the sensory posterior roots. After reaching the cord, every sensory signal travels to two separate destinations. The first one being one branch of the sensory nerve terminates almost immediately in the gray matter of the cord and elicits a local segmental cord reflex, and other local effects. Another branch transmits signals to higher levels of the nervous system, to higher levels in the cord itself, to the brainstem, and even to the cerebral cortex. Aside from the sensory relay neurons, the other neurons are of two types: anterior motor neurons and interneurons. We'll look into both of these briefly. Here is a schematic that shows the entire process that is happening. So here you can see the sensory root that goes out and branches into two parts. One of them which synapses on an interneuron and the other on an anterior motor neuron. So first we'll look into the anterior motor neuron. Located in each segment of the anterior horn of the cord gray matter are several thousand neurons that are 50 to 100% larger than most of the others and are called anterior motor neurons. These give rise to the nerve fibers that leave the cord by way of the anterior roots and directly innervate the skeletal muscle fibers. The neurons are of two types: alpha motor neurons and gamma motor neurons. Let us look into detail to what exactly is different between the alpha motor neurons and the gamma motor neurons. The alpha motor neurons give rise to large type A or A alpha motor nerve fibers, averaging 14 micrometers in diameter. These fibers branch many times after they enter the muscle and innervate the large skeletal muscle fiber. Stimulation of a single alpha nerve fiber excites anywhere from 3 to several hundred skeletal muscle fibers, which is collectively called the motor unit. Next are the gamma motor neurons. Along with the alpha motor neurons, which excite contraction of the skeletal muscle fibers, about one half as many much smaller gamma motor neurons are located in the spinal cord anterior horns. These gamma motor neurons transmit impulses through much smaller type A gamma or A gamma motor fibers, averaging 5 micrometers in diameter, which in turn go to small special skeletal muscle fibers called the intrafusal fibers, shown in the figure that we will just look into. These fibers constitute the middle of the muscle spindle, which help to control basic muscle tone. So this is a schematic showing as the muscle spindle, and in this particular schematic, we can see both the alpha motor fibers and the gamma motor fibers ending at with the endings shown as the alpha motor ending and the gamma motor ending. And we can also appreciate that the interior of the muscle fibers have some special intrafusal fibers, which signal to the brain about the tone or the percentage of contraction that is present in the muscle. There is further extrafusal fibers that are present that envelop the intrafusal fibers, and the sensory 1A and 2 nerves that innervate the interior of the muscle. We'll look into these as well in a bit. Next, after talking about the anterior horn neurons, we'll now talk about the interneurons. Interneurons are present in all areas of the cord gray matter in the dorsal horns, the anterior horns, and the intermediate areas shown between them, which is shown right. Interneurons are shown right here as given in the schematic. These cells are about 30 times as numerous as the anterior motor neurons. They are small and highly excitable, often exhibiting spontaneous activity and capable of firing as rapidly as 1500 times per second. They have many interconnections with each other, and many of them also synapse directly with the anterior motor neurons as shown in this figure. So here we can see an interneuron firing and having a synapse with the anterior motor neuron. The interconnections among the interneurons and the anterior motor neurons are responsible for most of the integrative functions of the spinal cord. Only a few incoming sensory signals from the spinal nerves or signals from the brain terminate directly on the anterior motor neurons. Instead, almost all these signals are transmitted first through interneurons where they are appropriately processed. So after the interneurons essentially serve as messengers, and the signals that come via the sensory nervous system have to pass through these interneurons before being transferred to higher motor centers. Next, we'll look into multi-segmental connections from one spinal cord level to the other levels via the propriospinal fibers. More than half of all nerve fibers that ascend and descend in the spinal cord are propriospinal fibers. These fibers run from one segment of the cord to the another. In addition, as the sensory fibers enter the cord from the posterior cord roots, they bifurcate and branch both up and down the spinal cord. Some of the branches transmit signals to only a segment or two, while others transmit signals to many segments. These ascending and descending propriospinal fibers of the cord provide pathways for the multi-segmental reflexes, including reflexes that coordinate simultaneous movements in the forelimbs and the hindlimbs. Next, let us look into muscle sensory receptors, muscle spindles, and Golgi tendon organs, and their control in muscle and their roles in muscle control. Proper control of muscle function requires not only excitation of the muscle by the spinal cord anterior motor neurons, but also continuous feedback of sensory information from each muscle to the spinal cord. This indicates the functional status of each muscle at each instant. That is, what is the length of the muscle, what is its instantaneous tension, and how rapidly is it is its length or tension changing. To provide this information, the muscles and their tendons are supplied abundantly with two special types of sensory receptors: muscle spindles which are distributed throughout the belly of the muscle and send information to the nervous system about muscle length or rate of change of length. And Golgi tendon organs, which are located in the muscle tendons and transmit information about tendon tension, or rate of change of tension. Let us look into detail into the receptor function of the muscle spindle. First, we look into the structure and motor innervation of the muscle spindle. The organization of the muscle spindle is shown as in the figure, that we have already seen before. Each spindle is 3 to 10 millimeters long. It is built around 3 to 12 very small intrafusal muscle fibers that are pointed at the ends and attached to the glycocalyx of the surrounding large extrafusal skeletal muscle fibers. Glycocalyx, if you remember, is a layer of sugar molecules that are present on the cell membrane of various cells. This is the point of attachment of the intrafusal and the extrafusal skeletal muscle fibers. Each intrafusal muscle fiber is a very small skeletal muscle fiber. However, the center region of each of these fibers, that is the area midway between its two ends, has few or no actin and myosin filaments. And thus, without actin and myosin filaments, this particular part of the muscle fiber obviously does not contract, because as we have already seen in muscle contraction, actin and myosin play a major role. And hence, what exactly is the function of this particular section of the intrafusal muscle fiber? Therefore, this central portion does not contract when the ends do. Instead, it functions as a sensory receptor. The end portions that do contract are excited by small gamma motor nerve fibers that originate from small type A gamma motor neurons in the anterior horns of the spinal cord, as described earlier. These gamma motor nerve fibers are also called gamma efferent fibers, in contradiction to the large alpha efferent fibers or the type A alpha nerve fibers that innervate the extrafusal skeletal muscle. Now let us look in detail into the sensory innervation of the muscle spindle. The receptor portion of the muscle spindle is its central portion. In this particular area or segment, the intrafusal muscle fibers do not have myosin and actin contractile elements. They are stimulated by stretching of this mid portion of the spindle. Two types of sensory endings are found in this central receptor area of the muscle spindle. They are the primary ending and the secondary ending. First, let us look into the primary ending. In the center of the receptor area, a large sensory nerve fiber encircles the central portion of each intrafusal fiber, forming the so-called primary ending or annulospiral ending. This nerve fiber is a type 1A fiber averaging 17 micrometers in diameter, and it transmits sensory signals to the spinal cord at a velocity of 70 to 120 meter per second, as rapidly as any type of nerve fiber in the entire body. Next, let us look into the secondary end. Usually one, but sometimes two smaller sensory nerve fibers, which are known as the type 2 fibers, with an average diameter of 8 micrometers, innervate the receptor region on one or both sides of the primary ending, as shown in the figure right here. So here we can see the group 2 fibers that innervate this end. This sensory ending is called the secondary ending. Sometimes this particular group 2 ending encircles the intrafusal fibers in the same way that the type 1A fiber does, but often it spreads like branches on a bush and thus differentiate and innervate a larger portion of the muscle. Next, let us look into division of intrafusal fibers into nuclear pad and nuclear chain fibers, which are the dynamic and static responses of and looking into the dynamic and static responses of the muscle spindle. There are also two types of muscle spindle intrafusal fibers. The first is nuclear band muscle fibers with 1 to 3 per spindle, in which several muscle fiber nuclei are congregated in expanded bands in the central portion of the receptor area, and the second being nuclear chain fibers with 3 to 9 endings in one muscle spindle, which are about half as large in diameter and half as long as the nuclear back fibers and have nuclei aligned in a chain throughout the receptor area. The primary sensory nerve ending is excited by both the nuclear back intrafusal fibers and the nuclear chain fibers. Conversely, the secondary ending, the 8 micrometer sensory fiber, is usually excited only by nuclear chain fibers. Next, let us look into response of both the primary and the secondary endings to the length of the receptor. That is the static response. When the receptor portion of the muscle spindle is stretched slowly, the number of impulses transmitted from both the primary and the secondary endings increases almost directly in proportion to the degree of stretching, and the endings continue to transmit these impulses for several minutes. This effect is called the static response of the spindle receptor, meaning simply that both the primary and the secondary signals continue to transmit their signals for at least several minutes if the muscle spindle itself remains stretched. Response of the primary ending, but not the secondary ending, to rate of change of receptor length, which is the dynamic response. When the length of the spindle receptor increases suddenly, the primary ending, but not the secondary ending, is stimulated especially powerfully. This excess stimulation of the primary ending is called the dynamic response, which means that the primary ending responds extremely actively to a rapid rate of change in spindle length. Even when the length of a spindle receptor increases only a fraction of a micrometer, if this increase occurs in a fraction of a second, that is, there is an increased rate of change of length of the receptor. Not only the change in length needs to be fast, but also the time that within which this change occurs also needs to be fast. The primary receptor transmits tremendous number of excess impulses to the large 17 micrometer sensory nerve fiber, but only while the length is actually changing or increasing. As soon as the length stops increasing, this extra rate of impulse discharge returns to the level of the much smaller static response that is still present in the signal. Conversely, when the spindle receptor shortens, that is, there is an exactly opposite sensory signal which occurs. Thus, the primary ending sends extremely strong, either positive or negative signals to the spinal cord to apprise it of any change in length of the spindle receptor. Hence, as we have seen already in the sensory nervous system lecture, there can be two types of receptors, one which adapt very fast and signal about the rate of change of the stimulus from the surroundings. And secondly, there are receptors which adapt slowly and inform the sensory nervous system of prolonged stimulation These also have counterparts in muscle spindles. By the static response that is generated by both primary and secondary endings, the muscle spindle informs the brain about the tone or contraction that is present. But while the dynamic response, that is the reaction of only the primary endings, which exhibit excess numbers of the action potential when there is a huge rate of change of muscle strength, tone, or contraction. This particular dynamic response gives the rate of change of muscles contraction and hence informs the brain of a stimulus that is changing rapidly instead of the normal static response. Next, let us look into muscle stretch reflex. The simplest manifestation of muscle spindle function is the muscle stretch reflex. Whenever a muscle is stretched suddenly, excitation of the spindles causes reflex contraction of the large skeletal muscle fibers of the stretched muscle and also of closely allied synergistic muscles. Let us look into the neuronal circuitry of the stretch reflex. The figure demonstrates the basic circuit of the muscle spindle stretch reflex, showing a type 1A proprioceptor nerve fiber originating in a muscle spindle and entering a dorsal root of the spinal cord. A branch of this fiber goes directly to the anterior horn of the cord gray matter and synapses with anterior motor neurons that send motor nerve fibers back to the same muscle from which the muscle spindle fiber originated. Thus, this is a monosynaptic pathway that allows a reflex signal to return with the shortest possible time delay back to the muscle after excitation of the spindle. Here we can see the whole muscle stretch reflex, where the sensory nerve, after a monosynaptic pathway, that is one synapse occurring, informs or signals to the motor nerve, which helps in the contraction of the muscle. Next, let us look into the dynamic stretch reflex and static stretch reflexes. The stretch reflex can be divided into two components. The first is the dynamic stretch reflex, and the second is the static stretch reflex. The dynamic stretch reflex is elicited by the potent dynamic signal transmitted from the primary sensory endings of the muscle spindles, caused by rapid stretch or unstretch or relaxation. That is, when a muscle is suddenly stretched, a signal, a strong signal is transmitted to the spinal cord. This causes an instantaneous, strong reflex contraction or decrease in contraction depending on the original stimulus of the same muscle from which the signal originated. The dynamic stretch reflex is over within a fraction of a second after the muscle has been stretched or unstretched to its new length. But then a weaker static stretch reflex continues for a prolonged period thereafter. This reflex is elicited by the continuous static receptor signals transmitted by both primary and secondary endings. The importance of the static stretch reflex is that it causes the degree of muscle contraction to remain reasonably constant, except when the person's nervous system specifically wills otherwise. Next, let us look into the Golgi tendon reflex. The Golgi tendon organ helps control muscle tension. The Golgi tendon organ is an encapsulated sensory receptor through which muscle tendon fibers pass. About 10 to 15 muscle fibers are usually connected to each Golgi tendon organ, and the organ is stimulated when this small bundle of muscle fibers is tensed by contracting or stretching the muscle. Thus, the major difference in excitation of the Golgi tendon organ versus the muscle spindle is that the spindle detects muscle length and changes the muscle length, whereas the tendon organ detects muscle tension as reflected by the tension in itself. Thus, the muscle spindle receptors that we had just discussed actually sense the muscle length and the stretching and unstretching of the muscle, whereas the tendon organ or the Golgi tendon organ determines the tension that is present and change and signals accordingly to the spinal cord. Transmission of impulses from the tendon organ into the central nervous system. Signals from the tendon organ are transmitted through large, rapidly conducting type 1B nerve fibers that average about 16 micrometers in diameter. These fibers, like those from the primary spindle endings, transmit signals both into local areas of the cord and after synapsing in the dorsal horn of the cord through long fiber pathways such as the spinocerebellar tracts into the cerebellum and through still other tracts to the cerebral cortex. The local cord signal excites a single inhibitory interneuron that inhibits the anterior motor neuron. This local circuit directly inhibits the individual muscle without affecting adjacent muscle, without affecting adjacent muscles. Next, we look into the inhibitory nature of the tendon reflex and its importance. This reflex is entirely inhibitory and thus it provides a negative feedback mechanism that prevents the development of too much tension on the muscle. Possible role of the tendon reflex to equalize contractile force among the muscle fibers. Another function of this reflex is to equalize contractile forces of the separate muscle fibers. That is, those fibers that exert excess tension become inhibited by the reflex, whereas those that exert too little tension become more excited because of absence of reflex inhibition. This spreads the muscle load over all the fibers and prevents damage in isolated areas of the muscle. After looking into the muscle spindle fibers, the Golgi tendon organs, and spinal cord reflexes, now we change gears and will discuss higher brain areas that control motor functions, especially looking into the cortex and cortical control of motor function. Thus, let us begin. Cortical control of motor function. Most voluntary movements initiated by the cerebral cortex are achieved when the cortex activates patterns of function stored in lower brain areas, that is, the cord, the brainstem, basal ganglia, and the cerebellum. These lower centers, in turn, send specific control signals to the muscles. For a few types of movements, however, the cortex has almost a direct pathway to the anterior motor neurons of the cord, bypassing some of the motor centers on the way. This is especially true for control of the fine dexterous movements of the fingers and hands. So here is a schematic that shows us the functional areas of the cerebral cortex, showing us the distinction between the motor areas and the sensory areas. The figure shown shown here shows us the functional areas of the cerebral cortex, anterior to the central cortical sulcus, occupying approximately the posterior one-third of the frontal lobes, is the motor cortex. The motor cortex itself is divided into three sub-areas, each of which has its own topographical representation of muscle groups and specific motor functions. These are the primary motor cortex, the premotor area, and the supplementary motor area. We will look into all of this in order to understand them in the next few slides. First, we look into the primary motor cortex. The primary motor cortex is shown in the first figure, that is this one, lies in the first convolution of the frontal lobes anterior to the central sulcus. It begins laterally in the sylvian fissure, spreads superiorly to the uppermost portion of the brain, and then digs deep into the longitudinal fissure. The figure lists the approximate topographical representation of the different muscle areas of the body in the primary motor cortex. Beginning with the face, this now we are talking about the second figure, that is right here. Here in the second figure, we are shown which part of the motor cortex responds or corresponds to the particular muscle that is present in our body. So here we can see that this particular part is corresponds to the face and mouth region near the sylvian fissure, followed by the eye and hand area, which are right here. In the mid portion of the primary cortex, the trunk near the apex of the brain, which is right here, and the leg and foot areas in the part of the primary motor cortex that dips into the longitudinal fissure, that is right here. This topographical organization is demonstrated even more graphically in the second figure, which shows degrees of representation of different muscle areas as mapped by Penfield and Rasmussen. This mapping was done by electrically stimulating the different areas of the motor cortex in human beings who were undergoing neurosurgical operations. Note that more than one half of the entire primary motor cortex is concerned with controlling the muscles of the hands and the muscles of speech. So this is the part we are talking about. A large portion, more than half of the primary motor cortex, only controls the muscles that are present in our face, mouth, and hands. Point stimulation in these hand and speech motor areas on rare occasion causes contraction of a single muscle. Most often, stimulation contracts a group of muscles instead. To express this in another way, excitation of a single motor cortex neuron usually excites a specific movement rather than a one specific muscle. So the question does arise of how exactly all of this was so minutely mapped by Penfield and Rasmussen. What happened is that Penfield, during neurosurgery of his patients, will, would take an electrical probe and would artificially stimulate the various areas of the motor cortex. When stimulated directly in the motor cortex, the electrical stimulation mimics a nervous signal, and hence the brain and the body feel as if a signal is actually being transmitted, and thus the person or the patient will show movement of muscle in the particular area which corresponds to the one stimulated by the electrical probe. Via meticulously analyzing various data from all neurosurgery patients, Penfield and Rasmussen were able to depict and map precisely what area of the brain deals with what muscles present in our body. One other important fact that we can note here is that various parts of the brain and the body are disproportionately represented. That is, the area of the brain that is being occupied by face muscles is obviously way larger than the normal face in proportion to the body. This is very nicely represented when all these different parts proportionately enlarged by the representation in the brain's motor cortex are put together in order to form a humanoid statue. This particular model is present in the British Museum and is known as the motor homunculus. A similar homunculus is present alongside the motor homunculus known as the sensory homunculus. And both of them nicely give us a taste of how exactly disproportionate the representation of different parts of our body are in our brain. I would encourage you to go and search for these motor and sensory homunculuses and look at the figure and compare it to the figure that we are seeing right here in this PowerPoint. Next, we move on to premotor area. The premotor area, as shown in this figure, lies 1 to 3 centimeters anterior to the primary motor cortex. The topographical organization of the premotor cortex is roughly the same as that of primary motor cortex. So here is the premotor cortex that is shown in pink around here, this portion. Nerve signals generated in the premotor area cause much more complex patterns of movement than the discrete patterns generated in the primary motor cortex, such as the pattern to position the shoulders and arms so that the hands are properly oriented to perform specific tasks. To achieve these results, the most anterior part of the premotor area first it develops a motor image of the total muscle movement that is about to be performed. Then, in the posterior premotor cortex, this image excites each successive pattern of the muscle activity required to achieve the image. The premotor cortex, basal ganglia, thalamus, and primary motor cortex constitute a complex overall system for the control of complex patterns of coordinated muscle activity. Finally, we look into the supplementary motor area that is shown right here. The supplementary motor area has yet another topographical organization for the control of motor function. It lies mainly in the longitudinal fissure but extends a few centimeters onto the superior frontal cortex. Contractions elicited contractions elicited by stimulating this area are often bilateral rather than unilateral. What this means is that stimulation frequently leads to bilateral grasping movements of both hands simultaneously, which might be required for climbing. That is, stimulation of the supplementary motor area essentially generates muscle activities which occur bilaterally or on both sides together. That is, one stimulation can generate contraction of the same muscles present in both of our arms. That is, these particular area might be useful for coordinated bilateral activities such as climbing. Next, we look into specialized areas of motor control found in the human motor cortex. Neurosurgeons have found a few highly specialized motor regions of the human cerebral cortex, as shown in the figure here, that control specific motor functions. These regions have been localized either by electrical stimulation or by noting the loss of motor function when destructive lesions occur in specific cortical areas. Some of the more important regions are discussed as follows. First one we'll look into is the Broca's area and speech. A premotor area labeled word formation, which is shown in blue right here, lying immediately anterior to the primary cortex and immediately above the sylvian fissure is called the Broca's area. Damage to it does not prevent a person from vocalizing, but it does make it impossible for the person to speak whole words rather than uncoordinated utterances or an occasional simple word such as a no or yes. Voluntary eye movement field in the premotor area immediately above the Broca's area is a locus for controlling voluntary eye movements, which is shown in yellow right here. Damage to this area prevents a person from voluntarily moving the eyes towards different objects. Instead, the eyes tend to lock involuntarily onto specific objects, an effect controlled by signals from the occipital visual cortex. This frontal area also controls eyelid movements such as blinking. Next is the head rotation area, slightly higher in the motor association area. Electrical stimulation elicits head rotation. This area is closely associated with the eye movement field. It directs the head towards different objects. The head rotation area is shown in pink. And next, the area for hand skills. In the premotor area, immediately anterior to the primary motor cortex for the hands and fingers is a region neurosurgeons have identified as important for hand skills. That is, when tumors or other lesions cause destruction in this area, hand movement become uncoordinated and non-purposeful. A condition called motor apraxia. The hand skill area is shown in orange right here. This was all we had for today. These are the references for the slides. The chapter 54, 55, and 56 from the textbook of Medical Physiology, Arthur C. Guyton, the 11th edition. I thank you for listening to this video.